So good morning, everyone. And today, in today's class, we will have a quick review of PCR, and then the actual objective is to learn about sequencing, right? So I think it will be a good way to just review PCR as well. It is simple. We already have discussed it. You have learned it before too. So the first thing is the objective of PCR, the polymerase chain reaction, is to have or clone multiple copies, make them several copies in vitro. That is not using bacteria or live cells, but in the ones. Of course, we still have to rely on the enzyme. We need Taq polymerase. And for the difference between the polymerization reaction is essentially almost similar what happens in vivo and in vitro. They need primers in vitro. They would need dntps, the enzyme DNA polymerase, and we need a template and magnesium ions, which will then be polymerized into a polynucleotide. So here we are, the differences are that the first thing is in vivo, when we do DNA is different, and there are ways to to denature the DNA, that is break the hydrogen bonds, and that is done by helicase. In here, we are going to use temperature or heat to denature the DNA, and that is a whole concept of it. We already have discussed, and we know the basic concept of polymerase chain reaction, right? What I wanted to discuss is that there are many different DNA polymerase. Often we discuss about the thermostable DNA polymerase. We just say it as TAC, that is identified from Thermophilus aquaticus. Because we are going to use temperature, we want a thermostable DNA polymerase. You can actually also use E. coli just like that if you are doing it at 37 degrees centigrade. But for denaturation, we are going to use high temperatures, something like that. And if E. coli polymerase was there, it would have been denatured. Okay. So I wanted to discuss about different types of DNA polymerases that are available and their advantages. So we should have to see that this is the Taq DNA polymerase that is often the one that is used for majority of the PCR reactions. But there are also many other types of DNA polymerases like PFU DNA polymerase, TLI DNA polymerase, TH and TFX, and many of them. So one thing we have to notice is something like the half-life at 95 degrees centigrade. Taq polymerase is about 40 minutes. I think it is not determined, or probably it's continuously stable, whereas TLI is half-life is 400 minutes if you put it at 95 degrees centigrade. And number of cycles also, you can see that. What we have to remember is speed and accuracy. Speed of polymerization and accuracy of polymerization, how faithfully the DNA is being copied. Right? That those are some of the differences. I think you would have learned about DNA polymerase Pol5 and Pol4, or UMUC UMUC and UMUD. So when the speed is very high, accuracy goes down. Okay. If you want high accuracy without any mistakes, then we have to compromise on the speed, right? And there is one more thing you need to think of. We will come to this again in a minute. I hope you know that DNA polymerases have the polymerase. Sorry, if this is 5' to 3', then we would have, say, 5' to 3'. If DNA polymerase was polymerizing here and there is a mistake there, the DNA polymerase has to move from 3' to 5' side. so that it can remove this the wrong mismatch that is there, and then it can repolymerize it, right? So this proofreading activity, that is moving 3' to 5' exonuclease activity, is not present in Taq DNA polymerase. That's why if there is a misincorporation of a base, it cannot repair it. It will just proceed ahead and it would have done that. But the other DNA polymerases, like PFU, for example, they have this proofreading activity as well. So their accuracy is usually very high. And you can compare the speed as well. Well, we will come to that. There is another 5' to 3' exonuclease activity. These proofreading ones do not have it, whereas thermophile Taq polymerase does. The number of errors, if you see between Taq polymerase and PFU, you can see that Taq polymerase makes more mistakes compared to that of PFU. And you can see the extension, that is the speed with which the polymerization can occur, nucleotides per second. Taq polymerase between 60 to 150 bases per minute, whereas PFU is only 60 bases per minute. So accuracy is high and speed is a little bit reduced. Okay, and we know from the TA cloning where we have learned that if you want to use TA cloning type, the 3' side DNA Taq polymerase usually leaves one base here, and that is the point that is made here. Yes, and others do not. Okay, so if we are trying to do a very long PCR, amplify a very long stretch of DNA, that means the probability of a mistake somewhere, if we are using Taq polymerase, is very high. I mean, relatively high, not very high. So one should be careful about the purpose, what you are doing. Are you cloning this particular fragment or amplifying this fragment for cloning purpose? And is it very long? If these are the cases, then it is better to use high fidelity DNA polymerases like PFU DNA polymerase. Okay, even though they might be slow, accuracy is everything. Say if we used, why did I say long? Is when you have longer DNA, the probability of a mutation is higher. And then if we clone that and we don't get the product, then the whole process is a waste. Right? So for those reasons, it is important that we learn that we use high fidelity DNA polymerases. Okay. I'm not going to discuss the PCR program. I will now go into sequencing. What is sequencing is simple from the name. We want to know the order of the gene of the nitrogenous bases that are present in a DNA molecule, right? All of bioinformation, if you call it, is about sequence of nucleotides, and the mRNA, the same thing, ribonucleotides, and protein is about the sequence of amino acids. The beauty of molecular biology or molecular genetics is about how the sequence of nucleotides is giving rise to sequence of amino acids, and that amino acid sequence is able to fold into a conformation, and that has a function, right? Therefore, we have to invariably know the sequence of nucleotides of any given DNA molecule, gene, or genetic element. There are two classical methods, and nowadays, of course, everything is done by NGS. We will discuss about NGS in some other class. Right now, we want to discuss, or we will discuss about maximum Gilbert method and Sanger's method. The reason why we have it here or why we still continue to read, is trying to understand the logic. This is not per se, you would never use, I'm sure you will not use maximum Gilbert method anywhere. Or maybe Sanger's method you will still use. But the point is, we learning the logic is the point of it. Okay. And the more logics we learn, the smarter we are, because we might be able to use those or similar logics somewhere else. That is one of the main reason why we are still studying these classical methods. So the maximum Gilbert method is called as the chemical cleavage method. I mean, we take DNA as the chemical cleavage suggests. We have different chemical methods. At that time, I think there was a race between maximum Gilbert, this group, and Sanger's group to come up with a very novel way of, as a way of sequencing DNA, because everybody by that time has realized that the sequence of nucleotide is the information that is running the show of life, right? So this was, and of course there was also a race for coming up with a method for sequencing amino acids. So this is a non-enzymatic method, and it is chemical cleavage method. So as the name suggests, chemical cleavage method is, there are specific ways about how we can, the chemical reactions were designed to cleave specifically at A or specifically at Z, G, and so on. And that is the basis of maximum Gilbert method. Let's see. So what we do is they take the sample, and this sample is made into single-stranded DNA. And if you remember, we learned about vectors. In that, we learned about single-strand DNA vectors. And one of the applications is involved in sequencing, right? The others being site-directed mutagenesis and for probes or blotting techniques, site-directed mutagenesis and probes as well. We will read about site-directed mutagenesis in the next classes. So here we have a single-stranded DNA, and we are going to use, we are going to add a radio label it. It is end labeling. So we would have used polynucleotide kinase. So on the 5' end, we will have the phosphate that is added here. And then we perform 4 different reactions. One is called a G reaction, the other one is called a reaction, and another one a T reaction, and another one C reaction. So the point here is, this is, one second. Here the reaction is A and G, and here the reaction is T and C. So these are purine reactions, and this one is for pyrimidine reaction. Okay. So, but whereas the other ones are G reaction and C reaction, they are specific for either G or C. The other reactions are purine reaction and pyrimidine reaction. You can call it that way. So we took multiple copies of these, and in one reaction we set up G reaction. That means everywhere there is G, there would be a cut. There is a G here, there is a G here, right? So you will have 3 different fragments. One of it where the cut might not have happened. These are not complete cuts, okay? And the other one is where the G has, the reaction is, I mean, the DNA is cut here, and another one where the DNA is cut here. So we have 3 fragments, one without any cut, like in this case, and one with a cut at the first G, and the other one, or this is the first G, because we'll always count from 5' to 3' here. So one reaction at first G, and the second cut at the the second G. Similarly, we have A and G reaction. That means in this reaction, everywhere there is A and G, there will be a cut. So as usual, with all the top lines here, we would not have any cut, so I will ignore it. So there will be a cut here because there is G here, right? That's why we have this. There will be A also will be cut. So we have the other fragment, and similarly we have these two fragments as well. So we are getting four different fragments because we have 1, 2, 3, 4. Okay, I didn't count. As I said, I am just ignoring the top one, uncut one. So the next reaction is pyrimidine reaction. That means everywhere there is T or C, there will be a cut. 1, 2, 3, 4, 5, 6, 7. Right? So you'll have 1, 2, 3, 4, 5, 6, 7. You'll have 7 cuts. And the last is the C reaction. We have 1, 2, 3. So we'll have these 3 fragments. So now it is simple because when we run this on a gel, okay, when we run this on a gel, they will run based on their molecular sizes. Of course, you have to use high density. I think it is polyacrylamide gels that are used, thin and high density, so that we will be able to separate them based on even one base difference. The difference between this molecule and this one is just one base, and we should be able to resolve those things. Say we have G, A and G, T and C reactions, right? How do you want to remember it? These are purine reactions, these are pyrimidine reactions, and you can put G and C. Often people, or including me, we get confused where we might write something like this. Okay. Instead, we should have had A and G, A and AG, and CT, and we might write like this. So the easiest way to remember is you have a purine reaction, you have pyrimidine reaction, G and C, and probably we'll be able to remember that. So if I have loaded these reactions, all of them will have the top strand, that is the uncleaved one. So I'm not discussing about it. So we have 3. One of it is at, I will just write for the sake of understanding, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I hope it is, how many are those? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 12. Okay, let's just put it like that. So in the G reaction, right, what we will see is there is a fragment at 3. It will give a band, and there should have been the other part of it. It will not give a band because that region does not have anything to be detected, whereas this part, it has the label, right? The 5' end, only the 5' end has the label. So it will get detected, whereas the other fragment will not get detected. 2, 3, 4, 5, 6, 7, 8, 9. So we'll have another fragment at 9. What does this say is that at position 3, we have, or say approximately around that, or 4, we have a G, and here we have a G. That is what it says. Now we will look at A and G reaction. So we are getting a 3 base fragment. We also have a 4. Then we have 3, 4, 5, 6, 7, 8 somewhere here, and we have a 9. I'm not going into all the details, so I'll not solve these two. Just we'll focus on this one. At position 4 or so, or position 4, assume at position 4, is it a G or A is a question. If we just focused on this reaction, the purine reaction, we don't know what it is. It is either A or G. And then we refer to the G reaction. And G reaction shows that there is a G. So it cannot be A, and it must be G, right? It's nice way of something like a puzzle. You're reading through, and while you're reading through, you are trying to figure out based on looking at data from other side. Let's look at this scenario. Here, it could be A or G. And the G reaction says there is no G. Then it must be A, right? So same way, you can figure out every base on all these cases, right? It is comparing two things and figuring out. That is the logical way of how we do stuff as humans. Majority of the animals can only contemplate on one issue. Whatever the data is here, that is what they can read. We have this concept, or the way of relative, the learning is about relative. We can determine something that is not very obvious by comparing it with something like, we can call it as a control here, right? And that is the reason why we still have this method in our syllabus, to know about, to be able to decipher the information. I hope you understood that. And in the next slide, we have the better illustration of it. So as I was discussing, the same way here, this is the gel diagram, and this is 3, you have a 9, 12, 12 is the complete one, so we'll just disregard that. Then we have with A and G reaction. So this is G because it shows here. This is A because there is no band here. This is A, no band. G because there is one, and the whole one. With with T and C, this is the purine reaction, and here is C, and this is the G. Yes. So let's do the thing faster. This is either T or C, but there is no C, so it is T. This is T, and this is C. T, T, C, C, right? And we can relate this sequence. So we'll start it with base numbers 1, 2, like that. So the first one is T, second is T, third is G, A, C, T, T, A, G, C, C. We are reading it from 5 prime to 3 prime. Why is that? Because the probe, the label, radio label that we added is on the 5 prime side. Only those fragments will get detected. And they would be, the smallest one with the label would be in the farthest down the gel. And so when we read the DNA, it should be from 5 prime to 3 prime. Okay. I think this is supposed to be a question, actually, where you should have been able to work it out. But today, I removed it. So if we had in the exams, you might get something like this. Either a sequence will be given to you, and you have to draw a gel diagram, or a gel diagram will be given to you and you have to do a sequence, right? Typically, it is not either of these cases, not just Maximum-Gilbert method alone. So I might give Maximum-Gilbert method and Sanger's method and ask you to draw the 2 gel diagrams for a given sequence. That is a quite common question that I usually have. So remember that for your next CIA. I hope you enjoyed it. And the best part of rDNA like course is, or if you're taking anything to a higher level of understanding, The goal is to learn the logics. And as I think I might have already told you, but I would repeat it. So this is us, and we are trying to seek knowledge. And as students, we are supposed to probably we are seeking knowledge, but we don't want the knowledge directly. We want to learn the ways of how to decipher or derive the most accurate, most possible accurate knowledge. And then our goal as an education system, right, is to learn the principles and logics of deriving knowledge, right. You might have a separate interest, or if I just told you the knowledge part and not the logic part of it, then you only know logic, but you cannot by yourself decipher the um, any other knowledge by yourselves in the future. So our goal is to learn these things, the ways of deriving knowledge. And at least in rDNA, my philosophy is about discussing about these techniques, logics, and other things, so you can derive any knowledge that you're reading from your research papers or follow other things. Okay, let's get to Sanger's DNA sequencing. It is called as dideoxy method, it is called as enzymatic method, and also chain termination method. It is called dideoxy method because of the ribose, right? CH2OH, and this is our 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So in a typical ribose would have OH and OH in second and third positions, whereas the ribose of the deoxyribose, we would say, would not have this, would not have the oxygen here, 2 prime H, that is ribose. This 3 prime OH has a central role in polymerization, right? The phosphate would be bound to this, and that's what make the phosphate backbone of nucleic acids. So when you say deoxy, it's understandable. That is, the 2 prime carbon is just having the hydrogen. And now we are saying dideoxy. That means we are having 2 OH, or we are just having a hydrogen here too. There is a deoxy here, and there is a deoxy here, dideoxy method. We are also going to use enzyme, that is again polymerase, and we call it as chain termination because the dideoxy does not have a 3 prime OH, so there will not be any further polymerization. So we are terminating the chain, and that's why we call that method as such. So this is deoxyribose, and this is the 2 prime 3 prime dideoxyribose, right? I think it's nice that they mention, or we should be mentioning it, because when you say deoxyribose, we didn't actually mention if it is 3 prime deoxyribose or 2 prime. What we find in DNA is 2 prime deoxyribose. Okay, so in typical polymerization reaction, we will have alpha, beta, and gamma phosphates, if you can see them clearly. And during polymerization, what happens is during polymerization reaction, the phosphates, beta and gamma, are removed or eliminated as pyrophosphate, which will further be hydrolyzed to inorganic phosphates. But they will not be incorporated into the DNA. If you see here, this alpha phosphate is the one that gets incorporated, and here we have a phosphodiester bond. and this 3' OH is so important or essential for polymerization to occur. But if we had a dideoxyribose like this one that is shown here, and if it gets incorporated, this is a dideoxyribose. If it gets incorporated, there is no 3' OH, so no further polymerization occurs, and this chain is terminated, right? So we can use this principle to for sequencing. I think Sanger was awarded Nobel Prize for this as well. He was also the one who has devised a method for protein sequencing also. I think he might have received.
Automated sequenators.
Yes. For protein sequencing too, he got, I think he got a Nobel Prize. Yes.
Yes, sir. Those were automated ones, sir. That's what he got Nobel Prize for, sir.
Yes. Automation was possible based on the Sanger's method. Anyway, that's the principle of it. So the idea is that we have a primer. This is the template, and there is a primer attached. And then we can use different dideoxy DNTPs or ATP, DD, CTP, DD, GTP, and DD, TTP, right? So if we have a primer and DNA polymerase is added, then it would add. And if any of these is included, say in this case, only the ATP is added. Anywhere there is A, this is T, right? So opposite to it or complementary, A should be added. In this location, we have the normal DATP or DNTP as well. All DNTPs are present as well, normal ones. And also in this reaction, we have added DATP, DDATP. So a normal one could be incorporated or a dideoxy ATP could be added. And if dideoxy ATP is added, the chain is terminated for this strand. For the other ones, the polymerization can keep on happening based on the template sequence here. And then if you keep going, and eventually again there is another T opposite to which T should be added. So what I hope you get the correct the point of it, that everywhere there is DATP, there is a possibility of DATP being added or DDATP being added. And if ATP is added, the chain will no longer grow. So using this, we can create a reaction. We can create a separate C reaction, separate G reaction, and separate T reaction. Just for memory's sake, I would want to ask, what are the reactions in Maxim Gilbert method? Anybody? I said a purine reaction, and I said pyrimidine reaction. Purine reactions would be A or G. Pyrimidine reactions,
C or T.
or C. Yes. And then we also have individual reactions. What are they? This would be,
We had individual reactions for individual nucleotides,
yeah.
and then we had combined ones. A,
We
G,
have G, and we have, what is
A.
this? C. Okay. In Maxim Gilbert method, we have, these are the reactions: G, AG, TC, C. Okay. You can remember based on purine reactions, pyrimidine reactions. In Sanger's method, we have it different. We have A reaction, G reaction, C reaction, and T reaction, separate for each of these nucleotides. Anyway, this is a Maxim Gilbert method is a degradation process, whereas this one, the Sanger's method, is a polymerization based one. Okay. We have performed, assume we have performed 4 different bases, 4 different reactions. We now have A reaction, G reaction, C reaction, and T reaction. And if we run the gel based on this sequence that is given here, we would get, say, A reaction will they will all separate based on their molecular sizes. And similarly with G, C, and T. And then we can figure out what is that base there. It must be G there because we see a band in G. This must be A because we see a band here, and T. This is much more straightforward in a way. You just have to keep going and say G, A, T, C, C, G, A, T, C, G. This is simple way of, this is simpler or less um, mental energy is required, I guess. So you can just write this sequence into this, and we will get a sequence that is from 5 prime to 3 prime read from the down bottom to the top, right? Read in this direction, 5 prime to 3 prime, and then we have a DNA sequence. Sanger's method is very useful, very simple also. And we can, this is how the gel usually look like. This is the um, sequencing sequence, Sanger's method of sequencing here. And I remember we have a topic called primer extension, and that we didn't discuss. At the time, I did tell you that it will go hand in hand with Sanger's method. So say, for example, I hope I'll be able to try to make it up. Let's see. This is a DNA molecule, okay? Single-stranded for ease of understanding. And assume it has coded for a protein, a mRNA as well. Upon expression, we have mRNA. Okay, this is the this is the 5 prime end of the mRNA, 3 prime end of the mRNA. And assume this is the open reading frame. I want to figure out, if we didn't know the sequence of we know the sequence of the template or the gene, okay, or the operon, you can count it as. This is the promoter, and then upon transcription, it would give mRNA something like this. How would I figure out the transcriptional start site of this mRNA? How can I figure it out? This we can do it in conjunction with the Sanger's method of sequencing. Say, for example, I will take a primer, and the primer is something like this. Okay, I will use the same primer for mRNA as well. Okay, here, the Sanger's method, here I have used a primer which is radio labeled, end labeled. So we will have it radio label on the 5 prime end of it. Now, what have I taken? Say I'm making 2 reactions, 2 separate ones. One of it is with the gene as template, or the single-stranded DNA, as I said. And we used a primer. Say we will call it as primer X, and it is radio labeled. And I am performing Sanger's method of sequencing. So something like this. What you see in this one is the Sanger's method of sequencing. So that is for the top one. For the bottom mRNA, I'm going to use reverse transcriptase so that the DNA will form. And until where will it go? It will stop at this end, right? It cannot reverse transcribe further than that because there is no template. Reverse transcriptase is a template dependent, or you can say RNA dependent DNA polymerase. So because where did mRNA stop? It stopped at here. And if we did reverse transcription, the cDNA that is formed will stop here. If we run the gel, okay, if we run the gel and see whatever this is corresponding to, right, whatever the end is corresponding to will also correspond to the base on the DNA. Can anybody, if you understood it, can anybody say why That is, I will rephrase the sentence so you can probably understand it again. The end of this mRNA here, the end of this mRNA here will correspond, if I run a gel like this, will correspond exactly to the base where transcription has started. Okay, can anybody explain why? The key is I'm using the same primer in both the cases. The length of the primer will go here to here. It might stop, and each of these is a basis, right? It will give the whole sequence as you can see here. And in this case, in the mRNA, the same primer is used, so it will base pair with the same uh, complementary sequence. And then upon production, it would stop here, which would mean that that is where the mRNA has ended, right? And that is what it indicates. The picture that I'm showing you is used for a different purpose, but it is still primer extension. There, what we were trying to do is find out the cleavage site. The mRNA is being cleaved. Okay, so we were trying to figure out where exactly the cleavages occur. So in some cases, you may have full length, in some cases, cleaved products. And we can use the same thing to map the cleavage sites as shown here. Okay, that's all right. You don't have to go through the process of it, the result. That's not important at this point. What is important is you understanding the logic of it. Okay, I will get back to primer extension sometime again, where I must be having the picture of the primer extension. Okay, but anyway, I hope you understood how a typical Sanger's sequence would look like. This is how it would look like, and you can read each basis. This is one base resolution. That's important. That's amazing. Sanger's method is easy for automation because we can use DNTPs. DDATP can be given one color, dideoxy, and with some kind of color. And each of these bases, DDG, DDT, and DDC, can be given one color. Say if we give green here, red here, blue, and yellow maybe. If we can give these colors, and so the PCR reaction is simple. You do all the PCR, you do PCR in one tube, all of them together, because now we have a way of differentiating them because each of these is labeled differently, right? The DDATP itself will have this green fluorophore, which can be excited, and then the fluorescence can be red. So when we perform PCR, we will get so many different types of fragments of different sizes. And then we pass it through capillary electrophoresis. It's a thin tube through which the sample is allowed to go. And then the DNA keeps on migrating. And here we have a laser here, which will keep exciting each of the bases that is passing by, each of the fragments that is passing by. And if the fragment has either any of these fluorophores, the fluorescent tags, they will be excited, they will fluoresce, which is detected by the detector and sent to the computer as a wave of intensity. If there is black, it means it's G. If there is, forget about this tagging here, we will follow this one here. If it is green, that means it is A. Black is G, blue is C, T is red is T, and so on. So the computer this is all automated. We can read, if we go by a sequencing like this, we can still read it, but then we will be able to read maybe 200 or 300 bases on a good day. if we go for automated sequencing, we can possibly read 800 base pairs, and it is much faster and less efforts, right? We don't have to do much. All we need to do is PCR, load the sample, and then the sequencing is done. Sanger's method is very easy for automation because of the individual reactions. It is based on PCR. So even if the template is very little, we can still get a lot of output from there. And the last part of it is, I think you also remember we discussed about primer walking. I hope you remember chromosome walking, the term. At that, when I was discussing about chromosome walking, I did bring up what is meant by primer walking. Firstly, what is chromosome walking? Chromosome has several genes or DNA sequence genes. And if we are trying to we made a gene library and we are trying to see, we know a sequence, we isolated a clone, used that as a probe, and if there is anything overlapping, that that could be identified using colony hybridization method. Now we can use the second fragment as probe, and perform colony hybridization and figure out which of the clones is having overlapping sequence like this. And thereby we can slowly walk through it, and we would know the order of the genetic elements that are present on the chromosome. We call it as chromosome walking. The primer walking is, the logic wise, it's the same thing. Only point is we know a small bit of sequence. We only know as initial sequence a little bit. We use that primer, we make primer based on that, and we will read about, say, 800 bases. Now we know the sequence of this part. So we can design new primers again based on this region, right? And then run the automated sequencing again, PCR sequence, Sanger sequencing, and we will know 800 more. Now use this region as a primer and keep on moving. That is called as primer walking. The difference between chromosome walking and primer walking is, in primer walking, we can find the sequence of it, the nucleotide sequence. Here we might not know the nucleotide sequence. We will only know which clone comes after which clone, and then we are able to make an ordered library based on chromosome walking. Yes? Okay, I'll stop here. If you have any questions, we can discuss. In the next classes, we will talk about a little more. I think we have to finish site-directed mutagenesis. We finished site-directed mutagenesis or not? We didn't. So we will finish site-directed. Yes, we'll finish site-directed mutagenesis, and then we'll also go with NGS at a later time. Little bit of NGS. We're not going to discuss a lot more. We need to quickly get to the. We also have a little bit to discuss about epigenetics a little, and we have to get to the 4th unit where we have to discuss some research papers and try to read more on them. CRISPR is also there. Okay. Any questions?
No, sir.
Okay.